reflect. To internalize it. Yeah, for sure, for sure. Yeah. I'm not a big skier, but did not seem optimistic anyway. Yeah. How far is it to get to good skiing from Eugene? Okay. Cool. Cool. So that's the life. Yeah. 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 There's lots of reasons in Oregon. Yeah. Yeah. I think pretty good. Yeah. Yeah. I think we're on a groove. Maybe we're like a little over diversified at the moment, but we have time to pull it back. So yeah. Yeah, I'm like looping in some NHP work now for the future. So yeah, I did my PhD in CPAT. So it's always been kind of like a long-term dream to come back. Now we've been doing. Yeah, yeah. I like to zag. Move into a field as it's dying. It's the way to go. Invigorate it. That's right. Okay, we'll give 1 more minute. Yeah, yeah. It's monocytes. Do you know phenocrenin? Do you know phenocrenin? She's new at Princeton. She's like a developmental geneticist, but she started this marmoset colony that's just like thriving. And there are all these opportunities for very cool, like sort of internally controlled experiments because they have twins. So you can kind of track, and so we do a lot of behavior tracking. So we're starting to like track the care asynchrony, yeah, yeah. We'll see. We'll see what happens with all this. Yeah, 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 yeah. I'm sort of irritated by how little NHP work there is in like subcortex and hypothalamus, the structures that I care about. We'll see. We'll see. I'm not putting all my chips over there, but a couple chips seems fun. All right, should we roll? You ready? Go. Okay. Good morning, Cosyne. Okay, welcome to the Neuroethology session. I was talking with a couple people just before, and we think this is the first Neuroethology session ever at Cosyne. Please correct me if I'm wrong. It's very, very cool. And to kick us off for the Neuroethology session is Cris Niell from the University of Oregon, speaking about expanding the parameter space of visual computation in mouse and octopus.
Well, thank you, and thank you all for coming this morning. It's great to be part of this first neuroethology session and giving you the opportunity to tell you about our work aimed at expanding the parameter space over which we study visual computation in the mouse and the octopus. So what do I mean by expanding the parameter space? Well, for comparison, consider how visual computation is studied in artificial neural networks. There's a couple of different dimensions that you can explore. For example, you can look at different neural architectures. For example, a simple feed-forward multi-layer network, a more complex convolutional network, or a transformer architecture. Likewise, you can vary the task domain in terms of the stimulus or training set and the objective. So for example, you can train a network to recognize handwritten digits, to recognize objects, or perhaps a more real-world task, such as parsing a moving traffic scene to control an autonomous vehicle. But in contrast, the way that we study vision in biological neural networks, i.e. brains, tends to be much more constrained experimentally. For example, a standard task involves a head-fixed subject looking at geometric stimuli on a screen. If you want to make it more dynamic, you can put the subject into a virtual reality environment. But there's a whole range of task domains that relate to things that you have to do to use your vision in the real world that haven't really been tapped into quite as much. For example, the ability to interpret and navigate through a three-dimensional environment, aspects of active sensing, how do you move your head and eyes to acquire information, as well as the wide range of visually guided actions that we can perform. Likewise, when it comes to studying brain architecture, in vision, we tend to focus on a relatively small subset of model species. For example, flies, zebrafish, and mammals. But there's a whole range of species out there with different brain organization, correspondingly different alternate neural architecture, as well as different visual capabilities that haven't been as well explored as these model species. So what I want to tell you about today is our work exploring these two axes. First, by expanding the task domain in a standard model species by looking at active vision in the mouse in the context of free movement and natural behavior. And then looking at a very different neural architecture by studying visual processing in the octopus brain. So I'll start off by talking about active vision in the mouse. And there's a really good reason why most studies of vision are actually performed in head-fixed or stationary subjects. And that's because when it comes to studying vision, it's very powerful to be able to present a controlled visual stimulus, typically on a computer monitor. And importantly, you know exactly what the subject is seeing, because it's right there in front of them. And then when you combine this with an operant readout of a decision, something like licking or moving a joystick, you have a very powerful approach to be able to study the processing and discrimination of specific visual features, as well as looking at things like learning and the cognitive aspects of decision making. However, these types of approaches are very different than the way that we use vision out in the real world, and actually remove many of the challenging aspects of vision. So in the real world, both the stimulus and the behaviors that we engage in are quite different. The visual stimulus that we receive tends to be dynamic and complex 3D environments. I really like this example from Mary Hayhoe's lab that shows how complex the scene is just from the point of view of a person walking down a rocky path. And of course, it's made more dynamic by the fact that both the head and eyes are moving. And likewise, out in the real world, rather than a single trial-based decision, visual behavior tends to be continuous and interactive. And importantly, it's coupled to the nature of the visual input. So for example, analyzing the size and texture of a rock to figure out if you can step on it, or analyzing the speed and trajectory of a moving object to be able to run and then intercept it. So these types of computations involved in both the visual processing and the behavioral output are much more challenging than these standard head-fixed discriminations. And likewise, these types of visual inputs and the regularities in the world are what our brains evolved to process. And these behavioral outputs are the types of things that our brains evolved to perform. So I would argue that it's likely that we're missing some of the major and most challenging computations that our brain has to solve simply because they aren't engaged in head-fixed paradigms. So this led us to want to develop tasks for the mouse that would tap into the types of things that an animal has to do in order to interact with its world in the ethological context that they evolved for. The first of these was developed by a postdoc in the lab, former postdoc in the lab, Jen Hoy, who wanted to study prey capture in the mouse. So she set up a paradigm where our standard lab mice could chase and pursue and eventually capture live cricket prey. You can see it here. Every time the mouse sees the cricket, it makes a beeline towards the cricket's location. Cricket escapes, it reorients, and makes another beeline approach. So even though we don't do prey capture out in the world on a regular basis as humans, these aspects of detecting a stimulus, orienting towards it, and approaching to intercept are key aspects of things that we do. For example, when we hit a tennis ball. So mice can do prey capture, but are they using vision to do this? And more importantly, is there an easy way for us to be able to quantify this? Because in contrast to a standard 2AFC task where you can calculate a psychometric curve and use signal detection theory, now we just have a mouse running around chasing a cricket. So we needed to figure out some ways to quantify this. Jen worked out a few different ones. This one here I like a lot. This shows the angle of the mouse's head relative to the cricket. How accurately is it aimed towards the cricket's location as a function of how far away it is from the cricket. And you can see that in the dark, the mouse essentially never orients towards the cricket's location, demonstrating that the mouse needs vision to be able to do this task, at least in this context. And then likewise, in the light, we can read off a couple of different values from this curve. For example, the distance at which they start approaching, as well as the accuracy of their orienting and pursuit. So this is kind of like a psychometric curve, but now in the context of natural behavior. So based on this, our lab and several others have gone on to look at the neural circuits that mediate these different components of the prey capture behavior. And I should mention that Jen has her own lab now at University of Nevada, Reno. Actually, she just got tenure recently. And she's studying the neural circuits that the development of the neural circuits for prey capture. So based on the success of the prey capture paradigm, we went on to develop another task to tap into something that animals have to do out in the world, distance estimation. This was developed by Phil Parker, another former postdoc in the lab. And he wanted to study distance estimation not based on stereo vision or the disparity between the two eyes like we often think of for depth perception, but based on motion parallax. So as you probably know, motion parallax is a phenomenon that as you move your head side to side, things that are far away in the world will appear to move less than things that are close up. And the nice thing about this is a computation that involves both movement and visual information for a specific computation. You take the ratio of the two and you get distance. So Phil wanted to set up a natural behavior to test this, and inspired by some old experiments by Mill Goodale and Mongolian gerbils, he trained mice to jump across gaps of different distances. You can see that mice are actually pretty good at jumping across gaps of pretty impressive distances. And what you might have noticed is that right before the mouse jumps, it bobs its head up and down. And that's what Mel Goodale had suggested generates the cue for motion parallax. So Phil, like Jen, wanted to go in and quantify this, and particularly he wanted to see whether mice could do this using just monocular cues, such as motion parallax, or if they need stereo vision. So he did the simple test of just covering one eye. This figure shows the success rate of jumping versus the gap distance. And you can see that they perform almost as well with one eye as they do with both eyes. So they don't need stereo vision to be able to do this. He then went on, and in order to analyze these active sampling head bob movements, he fit an AR-HMM to the behavior, pulled out those sampling movements, and showed that when the animal had one eye covered, those sampling movements increased in frequency, consistent with an increased need to generate motion parallax. So I really like this task because it uses engages active vision in two different ways. On the one hand, the animal has to move to be able to generate and acquire the sensory information it needs. And then on the output side, it's using that visual information for something that it actually evolved for, to be able to generate a force, the appropriate force to jump across that gap. And Phil now has his own lab at Rutgers where he's studying those two different aspects of the jumping behavior. So now we had a couple of different tasks where the animal is freely moving and could that tap into specific visual computations that we wanted to study. But you can see that there's probably a challenge in being able to study the visual processing that's underlying this. And in particular, the standard thing that we do in visual neuroscience, you present a stimulus on the screen, you record neural activity, and then you relate the two to each other through receptive fields or encoding models and so on. But in this case, the experimenter, us, is no longer in control of the visual stimulus. It's just the physical world. And of course, the mouse can move and look anywhere it wants within that visual scene. So we needed a way to figure out what the actual visual input is that the animal is receiving, what they're seeing, in terms of the input that's landing on the retina. So Phil set up a system based on some miniature head-mounted cameras to be able to figure out what the animal is actually seeing at any given time. One of these cameras looks out at the world like a little GoPro, so we can see the scene from the animal's perspective. The other camera looks back at the eye, so we can see where the eye is pointed within that scene. We have an IMU to measure head movements. We have a silicon probe to measure neural activity. And then we can put this all together and let an animal freely explore through a three-dimensional environment. So this is what it looks like in action. This is the visual scene from the world camera. This is the mouse's eye movements. You can see they become much more active once the animal's moving. A trace of eye position, head position, and the neural activity here. So you can probably already see that once the animal starts moving here, the pattern of neural activity becomes much more dynamic. And to some extent, that's just a consequence of the fact that once the animal's moving, the input's more dynamic, and that's reflected in the activity. But I do like to point out that this is what the field of visual neuroscience, what we've been studying for 50 years now, whereas this is what's actually going on in an animal's brain when it's moving through and interacting with its environment. So this was kind of our dream data set. We can see what the mouse is seeing, we can see where the eye is pointed, but there's actually two big computational challenges we needed to solve in order to make sense of this data. One of which is we need to take this position of the eye and figure out how to shift this world camera image to approximate what's actually landing on the retina based on where the eye is pointed within that scene. And then the second is to be able to relate that corrected visual scene to the neural activity through something like an encoding model and estimating receptive fields. So a former grad student in the lab, Elliot Abbott, took on this challenge. Elliot's now a postdoc up at UW with Bing Brunton. And he came up with an approach to be able to solve these two problems together, calibrating the eye tracking to get the shifted appropriate retinal input, and then estimating receptive fields. And he did this with a set of two small networks, one of which learns to take in the position of the eye, information about the position of the eye, and then learns the appropriate shift of the WorldCam image to approximate what's landing on the retina. And then the second is just a two-layer network, which functions as a GLM to predict the firing rate, predict the firing of the population of neurons. And importantly, the weights of this GLM end up being the spatial receptive field. So by training this whole thing end to end, it learns the appropriate calibration for the eye tracking to shift this image such that we can estimate receptive fields and predict neural activity accurately. So when we do this, sure enough, we get out receptive fields, much like what you might expect, both from our previous recordings in mouse, and even going back to Hubel and Weasel. So the receptive fields are shown here, color-coded, so that a region that responded to off or dark is in blue, light is in red. And you can see this receptive field as an elongated dark spot, an edge between light and dark, or Gabor-type receptive fields. So I mentioned that these are similar to what we had seen previously in mouse, but to test this directly, Phil and Elliot did the direct experiment of head fixing an animal, presenting white noise, and using standard methods to fit receptive fields, and then moved the animal into the arena, recorded the same units, and mapped the receptive fields using the approach I previously described during free movement. I should mention that it's only about a subset of neurons, about a third of the neurons that had receptive fields in both conditions, but for those that did, they generally matched up with each other quite well. So you can see they have similar on and off subregions, orientation, and so on. And if we calculate the correlation coefficient at the pixel-wise level, we actually get pretty good correlation coefficients with a mean of about 0.3, quite different from the shuffle distribution shown here. So on the one hand, this is quite gratifying. It shows us that we weren't just kind of pulling some kind of artifact out of our analysis. It also tells us that Hubel and Weasel weren't wrong, that the receptive fields mapped during free movement are similar to those that have been described previously. What for me is exciting about this is the possibilities that it opens up. For one, we can look at those neurons that were responding during free movement but not head fixation and try and figure out what types of information they're encoding. We can also go back to the behaviors I described previously and now record neural activity during those behaviors and know what the stimulus is. So for example, we can look and see how the cricket is encoded versus the background. Or likewise, we can look at aspects of the behavioral state. What's different in the processing when the animal is just passively exploring its environment versus actively hunting the cricket. And then finally, we can go in and look at aspects of visual processing that simply aren't present in a head-fixed animal looking at two-dimensional stimulus, such as things to do with movement through the three-dimensional structure of the world, or the active sampling movements, how an animal moves its head and eyes to be able to acquire information. And that's what I want to tell you about next. So we wanted to look in and see how processing in V1 depends on how an animal moves its head and eyes. So previously we had done some eye tracking during prey capture that I'm not going to go too deep into today, except to give you the takeaway, which is that we found that when an animal moves its head side to side, or as it sweeps it, looking around for the cricket or just moving through the environment, the eyes can make one of two types of movement. The eyes can make one of two types of movements to go with that head movement. They can either move in the opposite direction, so as the head moves one way, the eyes move the opposite way, which would stabilize the gaze at one point in space, or the eyes can make an abrupt jump in the same direction that the head is moving to shift the gaze to a new location. And together these create a series of stabilized images of the visual scene interspersed by these abrupt saccadic shifts. You can also see this in our corrected world cam video here. This is estimating the retinal input. And even though the mouse is moving its head through the world continuously, the visual scene is broken up into these stabilized periods of stabilized scene interspersed by the abrupt jumps each time the mouse makes the saccadic gaze shift. And of course, this is also true in humans. If I scan my head across the audience here, my eyes will compensate to fixate on one point and then intermittently jump to a new point. So we wanted to go in and see what the neural processing was corresponding to these two different types of eye movements. And in particular, there had been a couple of groups that had shown signals related to head movement in V1 of freely moving rodents. But we wondered whether that was due to the head movements itself or the corresponding eye movements and perhaps a change in visual input that's coming with that. So Phil, along with two outstanding undergrads, Emmeline and Dylan, who are both now in grad school, went in and recorded in V1 while a mouse just actively explored through a three-dimensional environment. No tasks, they were just freely exploring. They took every time that the mouse moved its head and broke it up into whether the eyes had made one of these saccadic gaze shifts or whether the eyes were compensating to keep the visual field stationary. No change in input. This shows one example neuron. You can see that this neuron fired briskly immediately after one of these gaze shifting movements, but no change in firing following the compensatory movement. We saw a diverse set of response types. Some neurons had a biphasic response, some had a negative response. But across the population, the response was predominantly following these gaze shifting head and eye movements that provide new visual input, as opposed to the compensatory movements, which were stabilizing the input. So this tells us that not all head movements are the same. It depends what the eyes are doing at the time, and in particular, whether they're shifting to acquire new input. So we saw this kind of diverse range of response types, but when we sorted them based on the latency to their peak positive response, we saw that in contrast to what looks like discrete types, these actually formed a continuum. They formed a temporal sequence from the neurons that responded immediately after the gaze shift to ones that went negative and then positive, and then the ones that just had a negative response. So there's this temporal sequence that gets set off after each gaze shift. You can actually see this in individual recordings. Each of these triangles here is the time of a gaze shift, and after each one, you can see this little ripple of activity going across the population. So each gaze shift that leads to new visual input is creating this temporal sequence, which suggested to us that it might be visually driven by that new input arriving after the gaze shift. So to test whether it was visually dependent, Phil and Dylan and Emmeline did the direct test of just putting the animal in the light, recording the response following gaze shifts. We see that same temporal sequence, and then put the animal in the dark and record from the same units. So these are matched units on these two sides. And you can see that while there's a small population of neurons that still respond in the dark, we think these are the ones that are actually encoding the head and eye movement, the motor corollary discharge. Most of the units are just reduced to a weak suppression, which we think is consistent with surround suppression, with saccade suppression. But overall, this temporal sequence of activation has been largely eliminated, suggesting that it's due to the visual input coming in. So if this is the result of the input, what are these neurons actually encoding? So to test this, we went back to standard classical stimuli to relate this back to what is known about typical tuning properties of neurons in V1. By first recording from the animal during free movement, getting the temporal sequence for each neuron, and then head fixing the animal and recording the same units in response to a classical stimulus, sinusoidal gratings. And in particular, we were varying the spatial frequency of the gratings. Spatial frequency is how broad the gratings are, so low spatial frequency is broad, high spatial frequency is the fine gratings. And when we plotted the preferred spatial frequency of the peak spatial frequency of each neuron as a function of its latency following the gaze shift, you can see this nice progression from the early neurons responding to the lowest spatial frequencies and the later responding neurons responding to the high spatial frequencies. So they're going from processing broad stimuli to the more fine stimuli. And this is consistent with the principle known as coarse-defined processing, whereby you first process the broad aspects of the scene and then fill in the fine details. And this has a lot of support from theory and human psychophysics, as well as some ePhys recordings. But our results show that this sequence from coarse to fine is kicked off every time the animal shifts its gaze to look at a new location. Every time it acquires new visual input, the temporal sequence goes into action. So you might think that maybe this is just a mouse thing, something about how they move their head and eyes. But it turned out that around the same time, Jake Yates and Jude Mitchell were recording neurons in foveal V1 of marmosets that were freely gazing on natural images. And they were seeing some unusual responses following the saccades. So we teamed up and analyzed the data in the same way. And sure enough, they saw the same temporal sequence following each saccade. And they were also measuring spatial frequency, and they saw the same progression from low to high spatial frequency as well. So together this shows that mouse and marmoset, even though they have very different ecological niches, different visual systems, foveal versus non-foveal, different repertoire of head and eye movements, in both cases, when you cast it in terms of what happens when the animal shifts its gaze to get new visual input, in both cases there's this temporal sequence and the computational principle of coarse-defined processing kicks in. So to wrap up this section, talking about vision and freely moving mice, I showed you that mice can perform various different natural behaviors that can reveal how visual information is both acquired and used. I showed you that we can implement freely moving recordings, combining electrophysiology with head-mounted cameras that allow us to perform measurements of visual coding even during active vision. And together, these two allow us to do the types of things that we thought we had to do head fix before, behavior and mapping receptive fields, but now we can do these during the natural condition of free movement. And then finally, I showed you how an aspect of active vision, these gaze shifts, actually format the visual input to precise temporal dynamics that elicit that principle of coarse-defined processing. So that wraps up the section on active vision of the mouse. Now I want to end exploring a different task domain, Now I want to switch over to exploring the other axis, looking at different neural architecture, in particular in the very different brain of the octopus. So the octopus is a great model to look at a radically different brain organization because they're a separate evolution of large brains that ended up with a vastly different organization. This figure here from Carrie Albertin's lab shows the number of neurons in the brain across the animal kingdom. And you can see that the cephalopods, including octopus, are this real outlier out here. They're very distantly related to us. They diverged, actually cephalopods themselves branched off from the rest of the animal kingdom over 500 million years ago, so shortly after the bilaterian split and the first emergence of real brains. And yet despite that, their nearest neighbors are the gastropods, so slugs and snails. But despite that, they ended up with brains that are several orders of magnitude larger than anything else in the animal kingdom besides the vertebrates. And because they had 500 million years on their own evolving their brains, they ended up with a very different brain organization, to the extent that it's almost like somebody handed you an alien brain and said, figure out how this thing works. So they're also a great model for studying vision because cephalopods have some remarkable visual capabilities. Some of these are shared across the animal kingdom, things like prey capture and navigation, whereas others are relatively unique to cephalopods. For example, many cephalopods can not only detect but discriminate stimuli based purely on the polarization angle of light, a property that we can't even really see. And of course, they also have the amazing ability to camouflage themselves by changing their skin patterning to match the visual scene around them. And they do all of this with a visual system that is in a couple ways similar to ours, but in most ways dramatically different. The similarity is primarily in their eyes. So the cephalopod eye is often given as a textbook example of convergent evolution, because even though our common ancestor had nothing resembling an eye, we both ended up with camera-type eyes, with a cornea, an iris, and a lens that focuses a high acuity image back on the retina onto the photoreceptors. But that's where the similarity ends. In contrast to the vertebrate retina, where there's a whole range of different cell types, bipolar cells, retinal ganglion cells, that do diverse types of computation, like we heard about from Greg last night, in the cephalopod retina, the only neurons are photoreceptors. The photoreceptors themselves fire action potentials and send axons into the brain. So it's just one synapse from photoreceptor to the brain. And then, of course, the brain organization itself is fundamentally different. Because we split so early in the evolution of brains, there's nothing that you can point to in the cephalopod brain that corresponds to the thalamus or the cortex and so on. In fact, the cephalopod brain consists of about 40 fused ganglia, the largest of which are these two optic lobes, which make up 2/3 of the brain. So 2/3 of their brain is devoted to vision. The optic lobes receive direct photoreceptor input and then send projections into the brain. This is shown in cross-section here. You can see the eye, the optic lobe between the eye and the central brain. As I mentioned, the optic lobe gets direct photoreceptor input, and then the output from the optic lobe goes to regions in the central brain involved in things like learning and memory, motor control, and so on. So to support those functions, it's thought that the optic lobe really performs the bulk of the visual processing that's happening in the cephalopod brain. If we zoom in on the structure of the optic lobe, shown here, the cell bodies are in green, surrounding neuropil in magenta. You can see that this also has an organization that's unlike anything in the animal kingdom or any other visual system. It has the outside of the optic lobe is these two cell body layers, the outer granular layer and inner granular layer, separated by a neuropil, which is where the photoreceptor axons come in. So there's this layered structure, which is unusual for an invertebrate, and actually kind of resembles the retina, the layered structure of the retina, but of course this is within the brain as opposed to within the eye. And then the bulk of the optic lobe is made up of this medulla. The medulla consists of these islands of cell bodies, and while they look like islands in this cross-section, in 3 dimensions they actually form a tree-like structure. But not a tree-like structure, for example, axons coming together into bundles, but a tree-like structure of cell bodies clumping together. If we then look at the morphology of neurons within the optic lobe, these are based on Golgi stains from J.Z. Young back in the 60s. Again, you can see that it's a very unusual organization with a wide range of different cell types, most of which aren't very familiar. There is one notable exception, which is that the outer granular layer here has cells that have an amacrine morphology, much like the amacrine cells of the retina, but again, within the brain instead. And then again, the bulk of the optic lobe is made up of this medulla, which has a range of neurons with diverse morphologies, but no real clear organization, except for the fact that a subset of them project axons out into the rest of the brain. So they mentioned this is where most of the visual processing is thought to occur. Photoreceptor input comes in, processed visual information comes out. Essentially, this is the entire visual hierarchy of the octopus brain. So all of its visual capabilities are based on the computations performed here. But we have almost no understanding of how this circuitry functions. And in large part, that's due to the fact that, in contrast, largely due to the fact that there's never been a recording of receptive fields within the optic lobe. We don't know what information they encode. And that's in contrast to, for example, the vertebrate brain, where even though there's a lot we're still learning, we know the basic pathways, what type of information is represented. Likewise for Drosophila. In the cephalopod brain, there's never been a recording of receptive fields beyond the photoreceptors. So we don't know how information is encoded, how it gets transformed throughout the circuitry of the optic lobe, and how it gets distributed to different brain regions that subserve those different behaviors. So this has been our goal, to figure out how this radically different visual system processes sensory information. And in particular, we want to figure out whether the optic lobe, the cephalopod brain, is implementing the same computations that we're used to seeing in other species, things like orientation, selectivity, invariant tuning, but just doing it with a different circuitry because it evolves separately. Or has the cephalopod brain come up with novel algorithms for being able to process the visual scene that might be unlike anything we've seen before, just because they had so much time to figure things out on their own. And then finally, we'd like to figure out the mechanism of some of these unique capabilities, that things like polarization, vision, and camouflage. How do you combine polarization channels? What does the receptive field for camouflage look like? So this was a project that was brought to the lab by a postdoc, Judith Pongor, who's now a staff scientist in the lab. This is the species that we're studying, Octopus bimaculoides. They grow to be about 5 centimeters across, so they're relatively small, and they're easy to raise in the lab. We actually study them when they're juveniles. So when they're about 2 months old, this is one of our subjects at feeding time, so you can see they can perform prey capture. At this point, their bodies are about 1 centimeter across, and octopuses only live to be about 1 year old. So 2 months old, you can kind of think about them as adolescents in terms of the full lifespan. At this point, all of their behavioral capabilities are intact. Being small just makes them a lot easier for us to work with experimentally. So over the years, several groups had tried to do electrophysiological recordings in the visual system, but had run into challenges. In particular, most of it had to do with the fact that these are marine invertebrates. They have very small neurons. There's some connective tissue that goes through the brain that makes them hard to isolate electrically. So we decided to take a calcium imaging approach. But even here, there's big challenges, because in the octopus, we basically don't have any genetic tools. We don't have things like Cris lines or Gal4 to express GCAMP. We don't even have viral tools, although some of us are starting to work on that now. But this means we don't have a way to express GCAMP, so we had to kind of go old school and actually directly inject a synthetic calcium indicator dye right into the brain. To do this, we have to do some dissection to get down to the brain and to be able to immobilize the animal. So this isn't like awake behaving prep like we're doing for the mouse. But we can immobilize the animal underneath the microscope, present visual stimuli on the side of the imaging chamber, and then look at corresponding neural activity. So if we do a small dye load and zoom in, we can get nice cellular resolution and see activity across populations of neurons. I should mention again, this is the first recordings within the cephalopod visual system. But more of what we've been doing actually to start off with is zooming out to look at the large-scale organization, figuring out what neurons in the optic lobe respond to and how this is organized both across the topography and the layers of the optic lobe. So we're zooming out, and a lot of the signal actually comes from the neuropill. So I think about the signals that we're measuring as more like population receptive fields or unsorted multi-unit activity. But what you can see is that every time this spot on the screen lands within this region here, a different region of the optic lobe lights up. So we can see visually evoked activity across the optic lobe. So I mentioned one of the first things we wanted to look for was the large-scale organization. And in particular, we wanted to look and see whether there was retinotopic organization. As you probably know, retinotopy is the idea that an image out in the world gets projected onto the back of the eye and then gets conveyed into a downstream brain area in an orderly manner, where each neuron has a spatially localized receptive field corresponding to one point out in the scene, and that these are arranged in an orderly manner to create a map of the outside world in that given brain region. Of course, retinotopic maps are very common across the animal kingdom, zebrafish, humans, many others. But there was some question as to whether there would be a retinotopic map in the cephalopod brain. And in particular, there doesn't seem to be a somatotopic map within the motor system. There's not a homunculus for the arms and the body. And that has suggested to people that maybe cephalopods don't use topographic organization, either because they evolved so separately and just never stumbled on that solution, or perhaps because they have a more distributed nervous system and so they don't use topography. So we wanted to go in and see if there was retinotopic organization in terms of spatially localized receptive fields and then a retinotopic map. So to do this, we mapped out population receptive fields using a sparse noise stimulus consisting of light and dark spots of different sizes, as well as light and dark full field flashes. We took the fluorescence trace at each location within the optic lobe. Remember, this is more of a population response in single units. But then we can take these traces and do reverse correlation to calculate a receptive field. When we do this, we get examples like this. So this is one location within one point in the optic lobe that responded to a light spot at this location, and then a nearby location in the optic lobe that responded to a dark spot at a nearby location. So these are the first measurements of receptive fields in the cephalopod visual system. They do have spatially localized receptive fields. They have both on and off responses. So now we wanted to go in and see how these are organized across the optic lobe. So we took the location of each receptive field that we had measured and color-coded it, and then placed it on the location that we recorded that receptive field. And you can see that there's this beautiful progression both along the vertical axis and along the horizontal axis. So there are indeed retinotopic maps in the cephalopod brain. We saw maps for both on and for off dark stimuli. But what you might notice is that most of the responses for the off stimuli were deeper in the optic lobe, in the medulla. And we think that's because in invertebrates, like cephalopods, the photoreceptors respond to light onset. And so you need to have a sign inversion to get that response to off stimuli, much like you have, for example, the bipolar synapse in the vertebrate retina. And so we think we've identified the location of that between the IGL and the medulla, and now we want to go and look and see what the mechanism is that generates that sign inversion. So we found two things that are shared: the retinotopic organization, the emergence of on and off pathways. But one thing that was different was we went in and looked at the response to those spots of different sizes, particularly for light versus dark. So this shows the response to a light spot of increasing sizes, 3, 6, 12 degrees, and full field flash. And you can see that as we increase the size of the stimulus for light stimuli, the response decreases. This is consistent with lateral inhibition or surround suppression, also common across many visual systems. But when we looked at the response to dark stimuli, the response to all three sizes of the spots was roughly equal. There is something interesting going on here with the response to the full field flash, or lack of response to full field flash. And we think that maybe has something to do with how this off response is generated, that perhaps you need edges between light and dark to generate the off response. But the net result of this is that the optic lobe in general is biased to being responding to smaller light stimuli as opposed to larger dark stimuli. And we wondered what might be causing this asymmetry. And it was actually Jose Manuel Alonso, who studies on and off pathways in the mammalian cortex, who suggested to us that it might have something to do with the statistics of the underwater scene. In particular, because water filters light, things that are far away are going to appear dark, such as landmarks or predators overhead, whereas things that are close up and small, like food that they might want to catch, would appear bright. Of course, that's an ethological explanation. As a visual physiologist, I'm also interested in understanding what generates these on and off responses and what's the mechanism that leads to that asymmetry in the size selectivity. So that's some of our results looking at the functional properties of the optic lobe. In the last couple of minutes here, I want to tell you a little bit about our work trying to get at the circuitry by looking at cell types. So I showed you this figure on the anatomy of the cell types. So we know a little bit about the morphological cell types, but we know almost nothing else about these cell types. We don't even know what neurotransmitters are being used by any of these cells, let alone the types of markers of cells that we're used to in other species, PV, VIP, and so on, that might eventually give us genetic access into the different cell types. So to address this, we did what's now the common approach of doing single-cell RNA sequencing. We take all the neurons and cluster them together based on their gene expression, and then we can go back into the brain and see where those genes and cell types are located. I should mention this was a big team effort led by a grad student, May Sonko, several other members of my lab and Andy Kern's lab, and I teamed up with Adam Miller and Andy Kern to make this all happen, including reassembling the genome, reannotating, and all of that. So long story short, we didn't find any cell types in the optic lobe that matched on to any cell types in other visual systems, consistent with the fact that this really is a uniquely evolved brain organization. Two things I do want to tell you about, the first of which is when we clustered the cells, classified the cells based on the neurotransmitter that they're using. You can see, for anybody who's used to the vertebrate visual system, this array of neurotransmitters is a little bit unusual. We saw a large population of cholinergic neurons, acetylcholine, glutamate, dopamine, and then these two populations that co-express both dopamine and glutamate. When we then went into the brain to see where these different cell types were located, we also saw a very unusual expression pattern. The dopaminergic neurons were pretty much all isolated to this outer granular layer. You might remember those are the ones that resemble amacrine cells in the retina. They have amacrine morphology. So it seems like there's a whole population of dopaminergic amacrine cells right at the input to the visual system. And then within the optic lobe, then within the medulla, the glutamatergic neurons and cholinergic neurons seem to be intercalated together in those cell body islands. So it's not like one island is glutamate, one island is acetylcholine. So again, this is a very unusual organization of these neurotransmitters. The last thing I want to tell you about is this population over here, which didn't express any neurotransmitter phenotype. But when we went in to look and see what genes they did express, we saw a number of early developmental genes, things like SOX2, DLX, which suggested to us that these might be neurons that are still undergoing development. And you can even see them kind of streaming up in gene expression space up to each of these mature populations, suggesting that these are immature neurons that are coming into the circuit. In retrospect, this actually makes a lot of sense, because it turns out that the cephalopod brain, including the octopus, grows throughout its lifetime, and actually grows exponentially, so roughly doubling in size every few weeks. So with that type of exponential growth, you need to have new neurons coming in to support that expansion. And so we think we've identified those neurons in this population here. We went in to see where those neurons were within the optic lobe. You can see that they're intercalating into the optic lobe, as opposed to, for example, building onto the outside. So they're actually filling in to this functional circuit. Remember, this is an octopus that's already out swimming around, using its vision to catch food and so on. And these new neurons are coming in. So I think this raises a couple of interesting computational questions. One of which is, how do you add units into a functioning circuit without disrupting that ongoing function? And second of all, what's the computational role of these new neurons? Are they just increasing compute because you have more processing units, or do they have something to do with either plasticity or memory storage? So with that, I'll wrap up the section on octopus vision. Hopefully I've given you an idea of how the weird biology of the octopus brain can give us some new viewpoints onto visual computations. I told you about our use of calcium imaging and cell type mapping to identify aspects that are both shared and divergent. Shared things being things like retinotopy, on-off pathway, divergent being the differences in size processing and cell type architecture, and then this surprising continual neural circuit expansion. So we're still in the very early days. These are broad strokes, but I think they provide the foundation for us now to be able to go back into this circuit and ask questions like, what's the tuning properties of individual neurons? How does that vary across cell types? How does that vary as you progress through the circuitry of the optic lobe? And eventually try and figure out what the computational architecture of this visual system might be, and what are the particular neural motifs that implement different types of computations. And then eventually, of course, link this up to the wide range of interesting behaviors that the octopus can do. So with that, I'll go to the most important slide, thanking all of the people who did this work, the current and past members of my lab, collaborators, and our funding sources. And happy to take questions.
Time for a few questions. Gord, you can go first.
Chris, that was over here. That was very cool. So obviously no GABAergic or glycine.
Yeah.
So is DLX tied into inhibition and is SOX2 tied into excitatory, or do you have any idea how that ranks out?
Yeah, there's a couple interesting things. I didn't go too deep in the molecular and development given this crowd, but since you're in the crowd, I will tell you a little bit about it. We actually identified 3 different markers that are shown over here that identify the main 3 lineages going out to the mature populations. I don't remember the exact how they mapped onto those specific SOX2 and DLX. But you can see that, and they were actually unusual neurons, unusual genes. There was the TNF receptor, myonurin, big brain, Interestingly, the whole immature population was marked by a gene that has no homology outside of cephalopods. So there's some gene that's giving rise to this unique immature population. And then finally, you asked about GABA, and we actually were a little bit concerned when we didn't see GABA in the brain, in the optic lobe to start with. It turns out it is there in other parts of the brain, but in invertebrates, both acetylcholine and glutamate can be inhibitory depending on the neurotransmitter expressed downstream. And in our sequencing, we actually found an anionic acetylcholine receptor, and so we think that maybe that's the inhibitory transmitter, and that acetylcholine and glutamate are playing that role of EI balance in the medulla.
In the receptive field comparison between movement and head fixed, I
Yeah.
noticed there were maybe 10% of neurons that had an inverted receptive field. Can you comment on those, please?
Yeah, to be honest, that I think is mostly just a effective. Say if you have a receptive field that has on and off, and they've shifted slightly relative to each other. So like if the camera isn't exactly, or the camera shift isn't accounted for perfectly, or if there's some small amount of shift between the head fixed mapping and the free moving mapping, that's basically going to put them into antiphase with each other. So yeah. That's really just...
You don't think you have a good enough control on the shift to say that these are, for example, prediction error neurons?
I wouldn't say that here. I think I'm a little wary of making too much about differences that occur. The fact that they're similar is, that can't happen by chance, but some differences could be if we aren't perfectly compensating. I do have a postdoc now who's looking actually across saccades and looking at how information is encoded across the saccades that might start getting at some of those questions.
You mentioned, I think that you found that roughly only 1/3 of the neurons in the mouse had receptive fields both in the active movement condition and in the passive viewing condition. Could you comment a bit more about that? Do you think that that's a technical problem and you just weren't able to align the moving visual fields well, or do you think that that might reflect some real differences in their visual response depending on input from other areas of the brain that might tell that neuron that the animal's moving versus still?
Yeah, no, that's a great question. Yeah, it was about 1/3 that had receptive fields in both. A lot of them had receptive fields in one condition or another, and I think some of that just has to do with the different spatial statistics. So, for example, we were using a white noise stimulus. It was frequency band limited, so it might not have been the right domain, to drive some sets of neurons during head fixation. We also were using we're just using a GLM, so any neurons that had a strongly nonlinear response might not get a receptive field. So I think there's a lot more that we could map there. But I think there is also a whole range of things that an animal encounters as it's moving through the world. So basically, the two stimulus domains are so different that I wouldn't have expected all of them to match up. And behavioral state is obviously one of those things as well. So besides the differences in the outside world, the differences in the inside world in those two cases, I could explain that. And it's definitely one of these things we want to explore, both better models to try and capture more of the neurons in both cases, as well as looking at the other factors. I should mention another thing that Elliot did when he was analyzing the data. He extended the model to be able to include head and eye position and tested to see whether they came in as additive or multiplicative factors, and they predominantly came in as multiplicative factors, so kind of like gain fields that you see in the monkey cortex.
Thank you.
Hi. Given the very simple receptive fields you found in the octopus, I'm wondering if you have any speculation on if and where visual object recognition is done in the octopus.
Yeah. So part of the reason I think those were simple is because we basically have a population receptive field. So you can imagine that within there are a whole bunch of neurons that were tuned to different orientations and they're averaging out. I'm not saying that that's what's happening, but we just don't have the resolution to do that. Right now, we're now starting to zoom in and do individual recordings of individual neurons. We definitely do see orientation selectivity there. It's kind of tricky to analyze because we have this 3D structure that we're taking different slices through. And so sometimes we see orientation selectivity, sometimes we don't. And we think it depends kind of on exactly how we're slicing through different layers. Kind of like if you imagine if you're recording from the retina and you took a weird slice, an oblique slice through it, sometimes you'd see on, you get bipolars in some, RGCs and others. So until we have more cellular resolution, potentially cell type markers, I wouldn't say anything too strong about that. We also aren't imaging real deep in the medulla, so the output kind of comes in from the deepest part of the medulla. I should mention, a lot of those diagrams I showed, I was kind of only showing the outer third so we could zoom in on that layered structure. But there's a whole, what one might think of as almost a hierarchy going down from that to the output from the optic lobe. And I would expect it's in those deeper parts of the medulla where you would see the things that correspond to higher up in the primate hierarchy, if they are there.
Maybe we can take one last question while the next speaker's setting up.
I was wondering about coarse-to-fine processing in the octopus. Do you see any evidence of coarse-to-fine? You know, like you had compared primate and the mouse. And do you see any waves in the octopus when you image that might be consistent with coarse-to-fine processing?
That's a great question. Oh, sorry. That's also a good question. One thing is we're doing imaging, which is slower, and most of that coarse to fine happened within about 100 milliseconds or so, or from 100 to 200 milliseconds following the gaze shift. So we might not see it with calcium imaging, but people are starting to work out electrophysiology as well, so we could potentially get to that. But octopuses do have very interesting eye movements. They make saccades. It's not clear that, like mouse, they may not necessarily be targeted saccades. They're more reset saccades associated with head movements. But they have things like OKR and VOR. They also use their vestibular system to stabilize because they can kind of move in all 3 dimensions, but they always keep their eyes level no matter how they're moving, which is potentially important for how they're processing the scene.
Thank you.
Let's thank Cris. Next, we're going to hear from Saikat Ray, who's going to talk about neural representations of real-world places and societies.
Hi. Our brains have evolved to work in the natural world, where you encounter many individuals, you encounter dynamic ecological settings. However, oftentimes when I feel like when I'm studying natural behavior, I'm in often slightly different settings. And this comes about often because of methodological constraints that we have when we're studying the brain to understand how it generates behavior, or for a need for natural experimental controls. So over the last few years, what I've tried to do is bridge this gap and try to understand the brain in a more ecologically valid and socially complex setting. To do this, I've worked with a remarkable group of people in the lab of Nachum Ulanovsky. And if you look at a graph of, let's say, social complexity and ecological validity, probably around 95% of all of classical neuroscience happens fairly close to the origin of this graph. So today I'll tell you a little bit about what we see when we move along the x-axis, so increase social complexity and see how do neuroscience in complex societies, and then move along the y-axis and see what happens when you do neuroscience outside in the real world in really ecologically valid settings. So I work with Egyptian fruit bats, and these are highly social animals. They live in groups, they make big colonies in caves, and essentially we started by asking a very simple question: how does the brain encode natural multi-individual settings? To do this, what we did was basically we caught a bunch of bats, and we set up a colony of bats in the lab where there were both male and female bats. We put them in there. They stayed there 24/7 for up to a year. There were no humans, and we had a bunch of ways to observe them. So we put a bunch of synchronized cameras. We had like a local GPS system to track where each and every individual animal was at all points of time. And we also had methods to do wireless neural recordings in them, which we did for a few hours every day. Now you might have sort of noticed I didn't mention what the animals were doing, because essentially there was no task in this experiment. The animals were just living their lives. And we were essentially observing a slice of it and trying to understand what's happening. So what do the animals do? They fly. They go from one side of the net to another. They would come and maybe say hi, go back. We had QR codes on them to track where they are. They interact in interactions. Sometimes they had a bit of a standoff. Sometimes they really went at each other. But usually at the end of the day, they came together and slept together in groups. So we started by looking at the hippocampus, which is a very conserved structure in all mammals. And if you do neural recordings from them when an animal is moving around in a relatively empty environment, what you find is a representation of locations. This is shown on the right, what's a place cell, what's classically known as a place cell. And essentially here you see the activity of the neuron, which is the high activity is shown in red, and this is known as a firing rate map. So what happens when we look at the neurons as the bats are flying in this room? So here again, you're seeing a top view of the room, and the gray lines that you see are the different flights that the bat takes from the top right corner to the bottom left corner in the room. Now, the red dots are the activity of the cells, so the spikes, and below is the firing rate map, so what you would observe. So it looks like the cell is only responding at a particular location in the hippocampus, and you would expect it's just a place cell. But what we see is that this cell sort of only responds like this when the bat is going to meet somebody. On the other hand, if you look at the same cell, when it's doing the same flights but going to be by itself, the cell does not respond at all. So there's something incredibly social about this. Now, could it be that, okay, this is happening because somehow the bat is flying differently when it's going to meet somebody versus being alone? So we controlled for all of these various factors which we know can affect spatial coding, like speed and spatial coverage and so on. And even after this, we find that remarkably 67% of all hippocampal cells, which is a fairly large fraction in this brain region, come out to be significantly social. Now then we asked, does it depend who the bat is going to meet? So we found that there were a lot of cells which actually encoded the identity of the individual. What does that mean? So over here you see that the bat, a neuron in a bat responds highly when it's going to meet bat O, but does not respond at all when it's going to, when the bat O is not present where it's going over. But it does not show such a differential response when it's going to meet a different bat, bat P, if you look at the same neuron, or bat 2. So there is not just spatial coding and social coding, but there's also identity coding on top of it, where you have encoding of individuals. But what's happening at other times during this whole period? The bats spend a lot of time on the nets where they hang around, and they're basically observing each other. So we reasoned, okay, we're in hippocampus. You might be representing a bunch of different things. It could be my own position, or your position, or my distance or direction towards you. And essentially with 4 or 5 animals, this quickly becomes a very multidimensional problem. So what we did was we used generalized additive models, which are, let's say, a bit more elegant cousins of GLMs, to understand which factors are contributing. And what you do is essentially you use all these behavioral variables of positions and directions and so on to predict the neural activity of each neuron. I think what was more interesting was we used this framework from game theory called Shapley values, where you basically run these models with all possible combinations of variables to understand, sort of get the best estimation of which factor is the neuron responding to. So in this example neurons, what we see is that, first of all, it encodes several animals together. The most important variable that this neuron represents is the direction towards bat 1, then the direction to bat 2, then the distance to bat 3. But also over here, you see that direction towards bat 1 is represented in a much more prominent manner. So there's identity coding going on here also when the bats are sort of just observing each other on the net. Now, the beauty of this experiment, or the lack of the experiment here, let's say, is that we are just observing these behaviors when, and they're all intermingled. So you are looking at the same neurons when the bats are flying, when they're on the net, and essentially you can ask the question, are the identities you're encoding when you're flying to meet somebody, is the same cell encoding the same individual when you're meeting them on the net? And that's what we find essentially. So let's say I'm encoding bat O while I'm flying to meet him and not the other bats. Then also on the net, when I'm just observing all the bats, then I most strongly respond to bat O. So we had this sort of invariance in identity coding which was preserved regardless of location, regardless of behavioral state in the hippocampal region, which we know very prominently as a spatial coding region. However, it's not just spatial coding. This sort of kind of reminds us a little bit of Jennifer Aniston cells, which was observed in humans like about 20 odd years ago. And essentially what they had over there was there were neurons which responded to Jennifer Aniston in this case, but not to other individuals, so there was identity coding. But there was also an invariance that it responded to the pictures in different orientations of Jennifer Aniston. And here again we see some single neurons showing invariance in identity coding, but this is the first time we see this in any other species. So overall, what I've showed you here is that the hippocampus in a social settings not just forms a spatial cognitive map, but more of a social spatial cognitive map. And I showed you about how you represent individuals invariantly, how you encode positions and distances of others. What I haven't showed you is that not only are you representing identities, you're representing really relevant social factors, like these neurons also encode the sex, the social hierarchy, and how friendly you are with other animals. And also they respond to specific social interaction events over here. But what about the real world? So to understand what happens out in the wild, we basically went to an island in the middle of the Indian Ocean, and we let our bats fly over there while we recorded from their brains. So this was quite a bit away from any landmass. And what we did was we went to Tanzania, which is on the east of Africa. We caught a bunch of bats from a cave. It's the same species of bats that we've been working with for several years. We did surgeries on them over there. Then we took a boat, we went to the island, we caught a bit of fish in the day, and in the night we did experiments while we let the bats fly outside, and we recorded the neural activity together with the positions out in the wild. Essentially what we did was we did 2 sets of experiments, and we could record lots of neurons and really get good quality data while doing this experiments in the wild. And I worked together with Saikat, who's a very talented graduate student in the lab, and we looked at 2 different brain regions in 2 different sets of bats. So we looked at hippocampus, again, where we want to understand how are place cells behaving outdoors in 3D, are there effects of altitude and stuff. And we recorded in a nearby brain region called the presubiculum, where we find head direction cells and where they were discovered. So today I'll talk to you a little bit about the head direction cells. So head direction cells basically fire when you're pointing towards a particular orientation in the room and moving in that direction. But it is unclear what happens in large environments. So does your head direction system shift in different parts of environments and only works as a local compass with sort of it shifting its orientation in different locations? Or can it work as a global compass and maintain its tuning throughout extended environments? So what we see here is like what you see here over is like the bat flying over several hundred meters. So on the x-axis about 250 meters, on the y-axis is like 350 meters. The gray lines are the trajectories, and the dots are now, again, the spikes and the responses of the neuron, but now they're colored by the direction when they fire where the bat was flying. So most of the dots are pink here, so the neuron was only firing when the bat was flying northwest. You can also divide up the environment into virtual little boxes and see what is the best tuning over there. And all of these boxes are, again, pink, showing that, again, the neural tuning in each of these small environments would have been essentially conserved in the northwest direction. So what we find is that it does work as a neural compass outside in the real world and over extended spatial scales. What's even more confusing about head direction cells is when you're in a lab, then usually what you have is an animal moving in a box, and there's a big cue card, much like the screens you have over here, let's say. And if these cue cards rotate, then the whole head direction tuning rotates with them. But this brings a conundrum. How can this work in the real world when you have like a big cue card in the sky which is moving throughout the night, the moon? So what we did was we recorded the same neurons, basically when the moon was still below the horizon and when it came up on the horizon. And we saw that it didn't make a difference to the firing of the neurons. So the system compensated for it and essentially maintained its tuning with or without the moon, unlike what you would see in a lab. And if you look at a population, essentially on the x-axis is the firing of the neurons, the direction they fired in with the moon, and on the y-axis when the moon was not present. And the fact that they all lie on this diagonal essentially shows that they do not change their tuning. So overall, when we moved the needle on exploring social complexity, we found that the hippocampus cognitive map is not just a spatial map, but in a social setting also works as a social spatial cognitive map. And we moved the needle on going into more ecologically valid setting. We found that head direction cells, which we know from the lab, can actually work as a neural compass in the real world. Thank you.
We have time for one, maybe 2 questions.
Do you think they have some sort of magnetic sense?
No, we tested for this. We actually recorded the head direction cells in the island, both outside and also inside in a room, and the tuning shifts. So they do not have a magnetic compass, because then we would expect the neurons to maintain the tuning indoors versus outdoors.
Thanks for the talk. I was wondering, these social neurons, do you see any reciprocity, or is, in terms of if bat1 has many neurons that are preferentially tuned to bat2, does bat2 show similar preferential tuning to bat1? And can you relate that to behavior? For example, if the two bats spend more time together than with other bats?
So what we do see is, in terms of tuning, that I'll answer the second part of the question first, which is that these identities sort of depend on who you are encoding, depends on some factors. For instance, you tend to encode males for some reason more, animals who are more dominant than you more, and animals who you are less friendly to more. Somebody I talked to in a conference told me, of course, you're encoding the asshole, so, you know. But apart from that, we don't really know if there's a reciprocity, and the primary reason for this is we were typically recording one animal at a time doing the neural recordings. We had some with two, but not enough to say anything conclusive.
I think we need to move on in the interest of time. Maybe you can catch Saikat at the coffee break. Thank you.
Hi, everyone. Today I'll talk about how walking fruit flies use directional memory in olfactory navigation. This is a collaborative project between the Abbott lab and the Ruta lab. And all the experimental work are done by I'll talk about are done by Andy Siliciano, Chad Morton, and Charlie Dowell. Yeah. Many animals rely on olfactory cues to navigate their environment. The classical model of insect odor navigation is a set of reflexive behaviors based on instantaneous cues, such as upwind search when encountering the odor and random search when losing the odor. We want to ask whether or how flies use memories of past odor encounters to guide plume navigation. And to address this question, the Ruta lab designed a virtual olfactory environment in which a half-fixed fly walks on a spherical treadmill. A wind tube rotates in closed loop with the flies heading relative to the treadmill, such that the wind appears to come from a constant direction in the allocentric reference frame. We can design odor plume of any spatial structure, meaning that we'll send appetitive odor through the wind tube to the fly based on its 2D location in the allocentric space. We reasoned that memory would be most useful in a stable olfactory environment, and therefore started with a stationary plume parallel to the wind direction. We see that flies ascend the plume by tracking along the air odor boundary. Upon entering the plume, they rapidly counterturn to exit, and once outside the plume, they perform the more circuitous exploration for tens of seconds before re-entering. We call this behavior edge tracking, and here we flipped all trajectory to the left side edge to visualize the density. We also find that this behavior cannot be attributed to random walk outside the plume. We compare the return outside bouts between random walk simulation and fly data, and see that random walk will require significantly more time and path length to achieve the same distance from edge, indicating that flies strategically leave and return to the edge. The edge appears to be a very salient landmark for flies during navigation. They are able to track edge directions 45 degrees tilted from the wind direction, or even orthogonal to the wind direction. This strategy allows them to advance along the plume when it's not aligned to the wind. They're able to adapt their trajectories to distinct plume geometries, traveling crosswind to enter the vertical plume and downwind to enter the horizontal plume. To better demonstrate these directional biases, we remove the plume after flies have edge tracked for 10 minutes and see that they continue to travel in the direction they previously used to return to the plume and maintain these biases for tens of seconds. Therefore, it seems like flies use memory of past odor encounters to bias subsequent navigation. To better understand the underlying computations, we designed a spatial state space model in which simulated flies alternate between leaving and returning states. The transition rates between these states depend on the presence and the absence of odor. We hypothesized that as a fly exits the plume, it updates an exit directional memory towards the current traveling direction by a small learning rate. It then uses this memory to bias subsequent velocity update in leaving state along with an additive noise term. Similarly, as the fly enters the plume, it updates an entry directional memory towards the current traveling direction by a small learning rate. And this memory is then used to bias subsequent velocity update in returning state with an additive noise term. We fit this model to data and run simulations with the learned parameters. Simulations show that the model captured the ability of flies to track various edge directions, showing here 2 example trajectories and about 20 trajectories plotted together for each direction. It also matches the summary statistics of fly data, including duration and range of inside and outside bouts. Our model suggests that we can change a fly's returning bias after operant training of its entry direction. To test this prediction, we presented flies with a plume consisting of 2 segments, tilted in opposite directions from the wind. We find that most flies continue to travel in the direction they previously used to return to the first segment, and therefore rarely re-encounter the second segment. Then we introduced an operant training session in which we turn odor back on whenever the flies spontaneously travel in the direction required to enter the second segment. Consistent with model simulations, we see that flies are significantly better at tracking the second segment after such operant training. These findings show that flies bias the returns to odor plumes using a directional memory that can be dynamically updated. We found that the central complex, evolutionarily conserved brain region in insects underlying spatial navigation, plays a very important role in edge tracking. In fruit fly central complex, the fruit fly central complex consists of the ellipsoid body, the perocerebral bridge, and the fan-shaped body. EPG neurons innervate the first 2 structures and represent allocentric heading as a bump of their population activity. The fan-shaped body is a grid-like organization with intersecting columnar neurons and tangential neurons. Columnar neurons integrate spatial signals such as heading and traveling direction and can represent the direction and length of a vector in the phase and amplitude of their population activity. Tangential neurons synapse onto a whole columnar neuron population and can modulate the amplitude of this population based on contextual signals such as olfactory input. We first confirmed that EPG neurons aligns with allocentric heading during edge tracking, and that silencing EPG neurons impairs their ability to track along the edge. We then asked whether components of our model, such as directional memories and state-dependent biases, could be implemented by neurons in the fan-shaped body. Analogous to many other anatomically inspired models of angular goal formation in the fly brain, we hypothesized that the directional memories during edge tracking are stored in the synapses from tangential neurons to columnar neurons. Memory updates towards traveling direction could be driven by dopaminergic neurons activated at each entry or exit. Distinct tangential neurons could signal mutually exclusive returning state and leaving state and gate the readout of these memories onto postsynaptic columnar neuron populations. These 2 populations then can converge to another columnar neuron population representing a go direction that provides a directional bias during navigation. While we currently cannot pinpoint the memory neurons in the fan-shaped body, recent work has found that FC2 neurons represent the fly's go direction when they try to travel in a constant direction to escape heat. And PFL neurons compares the fly's heading with this go direction to generate corrective steering commands. We therefore examined FC2 neurons to see if it can represent dynamically changing goals during edge tracking. We simultaneously recorded EPG neurons and FC2 neurons and examined their phases over time. We see that the two phases mostly aligned in the upwind direction when the fly is inside the odor and as it initially exits the plume. But FC2 phase starts to diverge from EPG phase and point in the returning direction before the fly turned toward the edge. Showing here is the average trajectory of example fly. We also look at the two phases as a function of time prior to plume entry for multiple flies and see that the divergence happens several seconds before the fly's return. Together, these results show that FC2 neurons seem to represent a state-dependent goal that could guide the fly's return to odor plumes. To summarize, we found that walking flies use the air odor boundary as a chemical signpost during navigation. They bias the returns to the plume with dynamically updated directional memories, and flies can use vector-based computations in the central complex for plume tracking, analogous to what ants and bees use during homing and during long-distance migration. And one of the future directions we're excited about is to leverage the robust behavior of edge tracking and try to locate directional memories in the fly brain. And with that, I'd like to thank my collaborators and advisors and acknowledge our funding sources. Thank you all for listening, and I'd be happy to take questions.
Thank you. We can take a question or two.
Hi, thank you. Really interesting talk. I have a very naive question. Why are they tracking the edge and not just going in the plume? How is that helpful, like, to define the source of the thing? Why is the edge more helpful than like just being directly in it and coming back in once you exit, if you exit randomly?
Yeah, we think that it helps them to progress along the plume towards the source. Sorry, when the edge direction do not align with the wind direction, shown here, you can see. Sorry, yeah. So these are relatively stationary plumes we're looking at, but in more natural environments, shifts in wind direction can cause a plume to meander and therefore no longer align with the wind direction. And the edge is the maxima of the air odor gradient and therefore can be very informative for the fly about information towards the source. Yeah, that's our intuition at this moment.
Okay, let's thank all of our speakers.
Hi, one quick announcement. If anybody arrived this morning and you haven't picked up your badge yet, please get it before noon. It's just at the other end of this of the hallway. Okay, thank you.